interested in genomics, there's a huge amount of opportunity to develop a subspecialty interest in this at the moment, particularly because they're offering funded MSCs in genomics in some parts of England to try and upskill clinicians with their genomic skills to become kind of experts in their areas. So it is something, and that's the pathway that I've come through to know a bit more about genomics. So it is something that you might want to have a think about. And there's certain areas within pediatrics where that's really, really relevant. Oncology would be the number one, but there are other subspecialty areas, again, where genomics is going to become fully integrated into those specialties. So this just demonstrates how much our knowledge of genetics has changed, even the short amount of time that I've been practicing. We used to think that all mutations cause disease, and we now know that there's lots of variation, which is completely normal. We used to say that all the areas of DNA that was non-coding was referred to as junk DNA, and we said it had no function. We now know that's completely untrue. Things you used to hear in clinical practice, it was really limited what testing we were able to do historically. So you might do a karyotype and a microarray as the kind of initial test that you would do on a baby, and you might say, okay, they're normal, there's no underlying genetic cause here. And again, we know that they're only the tip of the iceberg in terms of what we can do with genetic testing. And then moving on from that, even if we use a trio exome and we don't find any pathogenic or likely pathogenic variants, it doesn't mean there's no disease. And again, this is something you might come across in clinical practice, that, okay, we didn't find anything on our initial screening, but research is constantly changing what genetic variants we know about and our understanding of disease. So just because we don't rule it out, or just because we rule a genetic cause out at the time, doesn't mean that when knowledge increases, we might not be able to find something. So it's more about what we know for now than anything else. And that's all just in my really short time practicing, that those sort of changes and that understanding has really developed. So there's much more changes to come. This is going back in time a little bit more, but you probably remember learning about Mendel in school, but he really kind of picked up the kind of first set of rules that are still very applicable today for us understanding the influence of genetics and heritable characteristics. So in 1865, he investigated pea plants, and by crossing all these different pea plants with different characteristics, he basically determined that certain features are passed on and inherited between plants and their secondary generations. And some of these features are inherited in a dominant fashion, some in a recessive fashion. He also determined that during gamete formation, so the formation of the sex cells that you're going to then give a new germline to your offspring with, that these characteristics are kind of independently segregate. So you might get one or the other, and it depends, therefore you pass on. And all of this assortment of genetics is independent. So some move together, but some move independently, and there could be crossover and mix up of these characteristics. And all those rules still pertain today, but we have a lot of other knowledge that contextualizes that information. So that was way back in 1865. And then you've got a huge amount of progress since then. There was loads of work done in the 50s around understanding how these inherited characteristics, how these genetic traits are structured. So we start to understand the language of nucleotides and bases, and the A's and T's paired together and C's and G's paired together. And then in the 50s, Watson and Crick understood the double helix model that provides us with the structure of DNA. And then after that, we begin to understand more about what happens from the process of getting from DNA to what is a functional protein and an inherited characteristic that's determined by the function of the protein. And in the 90s, we started genome sequencing. This is more recent kind of genetic discoveries research and how it translates to clinical practice. So on the right there, you've got key genomic research projects. Sorry, the helicopter's taking off here, so it's really loud. But in the 90s, you had the Human Genome Project, which you've probably all heard of, but this was an amazing international collaborative effort to sequence the first ever human genome. So it had over 90% coverage, which was the most that is achievable at the time, because this wasn't using the next generation technologies that we understand today. This was using Sanger sequencing, which is really spelling out every individual base of the genetic code in a much more laborious process, because we now have next generation technologies which allow you to sequence lots at the one time, but this is really focused, really specific, and it was a huge international collaboration that spelled out essentially the first human genome. So that was an amazing work. And then in 2003, that was all finished. So starting in the 90s, finished in 2003. In 2012, you're now doing the 100,000 Genomes Project. So that's such a change in that 13 years, all that time taken to sequence one human genome. And then here we are sequencing 100,000 genomes in parallel. And this was 85,000 NHS patients who had rare disease which may have a genetic cause, or cancer, to try and get the most information possible out of that. And that was only possible going from that step from taking years to do one to being able to do 100,000, was because we have these next generation sequencing technologies which allow you to do so much at the one time, which we'll talk a little bit more about. But if you look over on the left, this is the translation of how this research is then moving into clinical practice. And again, it's really been quite amazing that really a short period of time between starting to sequence the human genome. In 97, you're able to use DNA sequencing in diagnostic services. So this was all single genes at this point. This wasn't doing any of the kind of whole genome stuff. But you're now able to say, okay, I think you have cystic fibrosis, let's look at your CFTR gene. But that's a relatively quick turnaround in terms of translation from research to what's happening in clinical medicine. And we now have the ability to sequence genomes of lots of patients who qualify for eligible indications. And that has been largely due to the 100,000 Genomes Project. So the 100,000 Genomes Project is research, but NHS patients were selected for it and they got information back. So there were clinical implications for NHS patients depending on the results of the project. And what happened is all that information that the patients contributed has also gone into a massive research database that is able to be accessed by researchers from all around the world to essentially generate new findings that potentially has prognostic implications, diagnostic implications, treatment implications that then feeds back into that system. So what I think genomics is an amazing example of is how research and clinical utility can operate hand in hand in a really effective way that is ethical, although there are various challenges which we can talk a bit more about when we talk about the neonatal side of things, and really kind of prop each other up, and both sides can benefit. So it is really exciting. And on the back of the 100,000 Genomes Project, we now have the NHS Genomic Medicine Service, because essentially what the project did was really develop infrastructure that allows us to provide genomic sequencing technologies to NHS patients as part of their normal integrated clinical care, because all of those labs were set up, all of those experts were there, were able to interpret things better, so they really have supported each other. So the NHS Genomic Medicine Service is an NHS resource, so it's a part of the NHS, it's not a part of the research side, that's all driven by Genomics England, but it's sort of developed in parallel and they use a lot of the same resources, but it means clinicians can routinely use genomic technologies for their patients. The focus is on patients who benefit the most, and that really means rare and inherited diseases and cancer. And it's delivered through genomic lab hubs, so that's the laboratory side of things, and that's all centralized because, as you can imagine, it's a certain number of patients who benefit from all around the country, but the cost of every single lab in every single hospital running these tests would be huge, so it's all centralized. So there's hubs that are expert centers in processing these tests, and genomic medicine centers, and again it's that concept of centralizing care, that is really highly specialized so that you get the best possible results, because there's a huge amount of responsibility that goes into consenting patients for the right tests, interpreting them correctly. And if we have experts all working together at one centre and centralise that care, it means that we get much better clinical care out of that. So that's the way things are structured. And if you want to read more about this, there's loads online about how the Genomic Medicine Service is delivered. And what they have is the National Genomic Test Directory. So you can go on, you can just Google that and access that. And you either look, depending on if your patient has a rare disease or cancer, you go on and look at the different directories, and you can look at all the clinical indications. So if you think you have someone who would benefit from genetic or genomic testing, you can go in and look at the indications and what tests they need. So it really is fantastic. The applications of genomics in healthcare is hugely widespread. Diagnostics is only a really tiny part of what genomics can add to healthcare. And that's essentially the process of doing genetic testing to come up with a diagnosis. So if you have a patient that you think has SMA, you would do a genetic test for that, and hopefully you would achieve a diagnosis. And there is also all the other whole genome and whole exome tests that we'll talk a little bit more about that have much broader scope in trying to achieve a diagnosis. It's also really important for screening. This is something that's kind of more in the research field at the moment, and we will talk a little bit more about that. But essentially with screening, what we are looking to do is identify a disease before it has become clinically apparent at a time point where we can intervene to improve outcomes with a specific treatment. And there's a huge possibility for genetic testing in this context. Family planning, genomics has already arrived and is fairly routine. You can do testing during a pregnancy for something if there's a risk of a genetic disease or if there are signs of a genetic disease. You can also do pre-implantation diagnosis, so you can go through the route of assisted conception. If you know there's a high risk you would have a child with a disease that could affect them in a way that is affects their quantity or quality of life. You can have embryos tested and only implanted if they have an absence of the gene that would cause disease. Precision medicine is amazing and is very much the way that healthcare is going. We're kind of trying to move away from you have X problems, so everybody gets X treatment, because there very much are genetic components to a disease that means that certain treatments would be better for different people, and we're going to talk a little bit more about that. It's also hugely important in infectious diseases. So we've used genetic technologies for a long time to even diagnose. If you're doing a PCR test, for example, to say whether a child has a bacterial meningitis or a virus, whatever it is that you're looking for, that is a type of genetic testing. But now we're using kind of these advanced technologies to find out more and more about infectious diseases, also to understand drug resistance, because it's the genome of the pathogen that affects how resistant it is or how susceptible it is to a medication. And if we can understand drug resistance, what we also have an amazing opportunity to do is change our drugs, develop drugs that mean that we can overcome those resistant profiles. So it really is excellent, quite amazing. This is talking a bit more about the kind of diagnostic side of things. You may be aware of this paper, but this is the Next Generation Children's Project, which did whole genome sequencing in children who were in NICU or PICU from a group in Cambridge, and looking at the clinical utility of offering them whole genome sequencing. So they achieved a diagnosis. These are patients who didn't have a diagnosis otherwise, in 21% of these patients. And of the neonatal cohort, 83% of the ones who had a genetic finding that directly influenced treatment, prognosis, or direction of care. So a huge amount of clinical utility there. And it was a rapid turnaround test, one of the first times that this had been applied in pediatrics, where you've got these sick patients and you need to know what's happening for them in an urgent way because they are unwell. And it was able to turn around the test in 2 to 3 weeks. And I think about 750 patients, maybe I'm getting confused. Let me have a wee look. 195, families participated in this. So it was really excellent. And it's now something that we do relatively routinely if you have these really sick children in PICU or NICU, and we think a genetic diagnosis is possible, that we're able to do a high turnaround of these really informative tests. And the other thing to say here is that it was relatively broad indications to get into this study. So actually, if we refine those a little bit, we can probably even get higher diagnostic clinical utility. And thinking about precision medicine, this was an excellent study, the PALO study, which developed a rapid point-of-care genotyping test to avoid aminoglycoside-induced ototoxicity in neonatal patients. So there is a specific mutation that if you have, it means you're incredibly sensitive to hearing loss if you're exposed to gentamicin or other aminoglycosides. It is irrespective of the level. So you may have hearing loss even if you never have a high level of gentamicin. So it is really beneficial if we could identify these patients prior to them ever getting aminoglycosides. So this study had 750 patients, and in that they found 3 patients with genetic variants that meant that if they'd been exposed to aminoglycosides, they would have been at risk of hearing loss, basically, and they were able to select different antibiotics for those patients based on this point of care test. So it takes 26 minutes, and it happens with a cheek swab. This is the gene drive set that does it. So this is the point of care kind of testing that will do it. And it's looking for this mtRNR1 mutation. So you take a swab from the cheek of your baby, you put it into the point of care testing. You can then go off and cannulate your baby, collect your bloods, get everything sorted. And 26 minutes later, you have an answer on whether they can get aminoglycosides or not. There was concern that this would lead to a delay in the administration of antibiotics, but actually their mean time to antibiotic administration essentially was unchanged between the before and after groups. So huge amount of possibility here, and it's recommended by NICE. Thinking about infectious diseases, this is a really nice paper looking at how much information can be derived from traditional genetic techniques that we would use to understand how an outbreak occurs, so how an outbreak evolves, and how organisms are changing in response to that outbreak, comparing those traditional techniques with essentially next generation techniques that gives you a lot more information. And essentially they found that they understood outbreaks in much more detail, including like from this water source to this, room in the hospital over to this patient. They could really kind of look at it in micro detail, step by step, and understand how organisms are changing in response to time with an outbreak, in response to antibiotics. So huge amount of possibility here. So I'm going to talk a bit about kind of key genetic concepts. I'm sure this is something that you will all remember from these kind of cobwebby recesses of your mind that did genetics in medical school, or if you've done your exams, you're probably a little bit fresher on this than maybe even I am. But just to refresh things for you, genomics is the study of an organism's complete set of genetic information. So it's sort of all encompassing, and it's also trying to understand how genes interact with each other and what influences expression and how things move forward. Genetics itself, when you use that term, is much more focused, and it's the study of specific genes. And when we talk about genes, what we're meaning are individual units of inheritance. So certain runs of DNA, certain sentences of the genetic code essentially derive a gene that goes on to encode a protein that has a functional implication and will determine a characteristic that is something that is heritable. So that's how we understand the difference between genomics, genetics, and genes, if that makes sense. Often the language is used interchangeably, especially when you're talking about genomics and genetics, but there are subtle differences in actually what these terms mean. And if we think about all these sequences of DNA, we have both coding and non-coding DNA. And when we talk about coding DNA, what we are talking about is sections of the genetic code that result in a functional protein at the end of it. So it's very kind of clear and linear that that genetic code eventually will equal that protein, and it's going to have a certain function. But the non-coding regions, we used to talk about them as junk DNA. We thought that they were just sort of these buffer regions, but actually they do have a huge amount of influence. Does anyone know what non-coding DNA does? Is there any answers in the chat, Martina?
There are no answers in the chat right now.
Okay, that's fine. Non-coding DNA has a huge amount of a regulatory role. So what it is doing is influencing how coding DNA is expressed, what happens to that that protein in that route of making it. So it still has clinical implications, and it's sort of just at the start that we are beginning to understand non-coding DNA. Where do you think the majority of disease is, in coding or non-coding?
Is it in the non-coding? I'm going to guess.
So it's in the coding. So only 1% of the genome is coding. By that, also the other term that we would use for that is the exome, is the coding regions of DNA. So only 1% of the genome is coding or exome DNA, but it accounts for 85% of disease in there. But the non-coding is this huge amount of the genetic code. So less cause for disease, but that may change over time as we research these areas of DNA more and understand more about them. But it's totally fascinating, those relationships and what this non-coding DNA does. So thinking about how all of this is structured, these genes, these runs of DNA that determine inheritable characteristics are packaged into chromosomes, and you're probably all aware of chromosomes. They exist in the nucleus. There's only one other place that's not in the nucleus where we have DNA that's really important. Does anyone know where that is?
Mitochondria.
Yeah, absolutely. Mitochondrial DNA is really important, but the rest of the DNA are all in the nucleus, and it's in the vast majority of cells. There's only some cells which don't have a nucleus and therefore don't contain that nuclear DNA. Does anyone know what those cells are? It's red cells and platelets are the ones that don't have that. But otherwise, all the other cells in the body, all the other somatic cells carry 46 chromosomes. So that's 22 pairs of autosomes, and they're your kind of regular chromosomes. And then you have a pair of special sex chromosomes that determine if you're XY, you're going to be a boy, if you're XX, you're going to be a girl. And then there's some other variations on that due to various genetic errors that are associated with disease, which I'm sure you're aware of. Your somatic cells have a complete set, so we call that diploid, and your sex cells, so that's your sperm or egg carrying cells. They carry half of the genetic information that you need, because you're then going to unite with another sex cell and create a complete set of genetic information. So that's in your gametes. You have what we call a haploid set of genetics. So here you have 23 chromosomes, 22 of these single chromosomes, so they're not paired, which gives you the 23. 22 are your regular, and then you have either an X or Y, and it's the combination when those two things meet that create a new person and give you your full set. When we talk about a locus, what we're talking about is a precise genetic location. So we might talk about the CFTR gene. It's at 7q31.2. That means it's on the long arm of chromosome 7 at location 31.2. That's the locus of the CFTR gene. And then it will span however many bases from there. When we talk about an allele, what we're talking about is a variant of the DNA sequence at a particular location, and we have 2 of them. So this is all going back to your P plants and the fact that you inherit half of your genetic code from your mother and half from your father. You end up with 2 alleles for each gene, so different versions of a gene that encodes the same thing. And a really good example, again, that you will recognize is that you have 2 alleles for your eye color. So if you have a big B and a little b, you've inherited from one parent an eye color brown allele, and from another parent an eye color blue allele. And because the brown allele is dominant, you will have brown eyes, even though you have different, two different coloured alleles. And it depends on how things are expressed, if they're dominant, if they're co-dominant, etc., what actually will clinically manifest. But we have two alleles of every gene, one from each package of our inherited characteristics. And then if you remember, DNA is only part of what gets us to that functional part, which is where we have a protein. We have to transcribe DNA to RNA, and then it's translated into a protein. And various things can happen along the way. That means that not all of your genome results in a functional protein. There are all sorts of other factors that determine what is transcribed or translated. So even if you have a certain amount of genomic information, if you have errors elsewhere in the process, you can end up with a different protein. So although this is focusing on genomics, it's kind of a whole series of things that we now have to look at. So understanding the genome is only part of it. You can also look at the transcriptome, and that is all of the RNA transcripts that are in a cell, and that's different in every cell. So if you look at a liver cell, the transcriptome will look totally different from a heart cell, for example, because they are phenotypically different cells, even though the genome in those cells is the same, because it's all about what's expressed, and the genome's doing all sorts of things, and all those regulatory regions are making all decisions so that you're getting the right phenotype for the right cell. But it's also errors that can happen at these different stages of transcription and translation that ends up with a different functional endpoint. When we talk about the proteome, we're talking about what has been transcribed from RNA to protein. So that's the sum of all the proteins that are present in the cell. And when we talk about the metabolome, that's all the small molecules that determine the biochemical environment of a cell. And that gives us the phenotype. And what you get is some really amazing work that various clinicians do, where they take different patients who have different diseases and they try and understand in a set number of cells. So if you're doing something, I heard of someone recently looking at the genome, transcriptome, proteome, and metabolome of a small number of patients who had chronic lung disease in the neonatal period when they were older. So they were looking at them kind of as young adults versus control patients. And you're trying to understand where these different things happen, because it may not be something that is going to happen to you from birth if it's not encoded in your genome, but all of these things can change and be influenced by the environment and your course through life. So it's totally fascinating, and all of that helps us understand disease and helps us therefore kind of work on new treatments and all of those things that happen downstream. But this level of understanding that you can get if you focus at all these different parts is just huge. Genetic variation distinguishes individuals, but not all of that genetic variation is disease. We all have thousands, hundreds of thousands of genetic variants that are different from the kind of the generic DNA template, and that's absolutely fine. That makes all of us our own people, and all that genetic variation, a lot of it is positive, but some genetic variation encodes disease, and that can be inherited. So something that you get at the point of your germline being established from the gametes of your parents coming together, or happen as mistakes down the line. So that can happen in replication, and you get replication to DNA as you're creating from a fertilized cell into a a fully formed human baby. So it can happen at those stages, or it can happen at later replication. So cancer is a really good example of that. It's not something that you've inherited as a mistake in your DNA in your germline, but you get these errors that accumulate, for example, with age. That means that you're more likely to have a cancer as the cells are growing out of control. Or there can be direct damage to the DNA template from things like carcinogens that really increase those processes, the risk of those processes. I've already talked a little bit about patterns of inheritance, and I've mentioned autosomal dominant and autosomal recessive, and that we owe that understanding to Mendel and his pea plants. Autosomal dominant means if you have one copy of a genetic variant that is pathogenic, you would expect disease. Autosomal recessive is that you have two copies. X-linked dominant is associated with the X chromosome, so it's dominant if you only need one variant on an X chromosome to be expressed. And generally here, what will happen is that it's often lethal in males, because they don't have the protective factor of the other X chromosome, and it's sort of milder in females, or they might be described as a carrier because they have the other allele. And X-linked recessive is a mutation on both genes of your X chromosome that leads to disease, and it's typically just males, because again, you don't have the two, so you only have the one, and you can't be protected in any way by the other one. So even though it's recessive, just because you have one, it will always be expressed, and it's much rarer in women to have two of those coming together. It almost never happens. Mitochondrial DNA is if you have mitochondrial errors, and they're passed on through the matrilineal lineage. So it's only the egg cytoplasm that contributes to the mitochondrial DNA that you inherit. So it all comes from your mother. De novo is when something occurs for the first time in the offspring and it's not there in the parents. But there are a huge amount of other genetic concepts that influence how disease is inherited. You can get variable penetrance. So that is when different people in the family might have the same mutation, but for whatever reason, it affects them very differently. So they may have a much more severe phenotype, or it affects them much earlier because the penetrance of that gene looks different based on the other genetic characteristics that you inherit or environmental factors. Anticipation is where something gets worse in successive generations, and that typically happens with genetic variants that are associated with a certain number of repeats, because you accumulate more and more repetitive errors over time. So something like the trinucleotide expansion disorders get worse in successive generations. Huntington's is one of them that you've probably heard of, and they show this anticipation of getting worse. Polygenic inheritance is where it's not just about that one gene, it's multiple genes and or their interaction with the environment that determine whether you have disease. And that is kind of true of things like diabetes, for example. It's very rare to have monogenic diabetes. It does exist. So that is your kind of like MODY patients. But most diabetes, for example, is polygenic, but you will have genetic risk factors that mean that you might see these families where they're all getting kind of young diabetes, and then it's the kind of other environmental factors and lifestyle factors which contribute as well. And then you get variation in regulatory regions and epigenetics. Epigenetics is totally fascinating. And that is essentially where you don't have any error in the genetic code itself, but you get different epigenetic setups that will essentially determine whether genes are expressed or not. And that can influence disease. So you get something called methylation that will tell you whether a gene is transcribed or not. It has an influence on whether genes are transcribed. And it's sort of, with methylation, you can cover genetic areas methylate them and make them essentially totally covered up so that those genes aren't expressed. But the actual genetic code hasn't changed. It's just that it's covered for a period and it influences expression. So it's really, really interesting. It is not just what we understand from school biology and Mendel. There's so much other stuff that influences what's expressed and how disease can occur. You also get a huge amount of different types of variants. So you can have a mutation in a single base. So we call that a single nucleotide variant. And generally, we're talking about substitutions when we talk about that. So rather than having an A, you might have a T, for example, at a specific area in the genome. And it is completely variable, the effect that that has. You might encode a different amino acid if you change the spelling in that genetic code. You might have the same amino acid, or what you might do is if you change the amino acid, it could be a stop. It could be a stop amino acid, so then you shorten your protein. So that has a huge impact. So it can have no impact, a small impact, or a huge impact if you have these single little spelling errors. We'll all have loads of these, and most of them don't cause disease, but there are some that can. Indels are insertions and deletions, and they cause a problem because we spell out our genetic code. Every 3 letters will essentially translate in the end to an amino acid that essentially will affect the function of the protein. With insertions and deletions, if you're doing anything, because we make those amino acids coded in each 3 bases equals one of those codes. If we have an insertion or deletion that's out with a multiple of 3, it changes everything that happens thereon, because it shifts. If you just put in 1, it then shifts the spelling that happens thereafter, because you are still going to read it in sets of 3. If you have indels, insertions or deletions that are numbers of 3, you just get an additional amino acid, and that can still have a big implication, but it's generally not as bad than if you shift everything that happens after to be different. And generally we talk about indels when they're less than 50 base pairs, and if they're more than 50 base pairs, we kind of talk about them as structural variants, because they're much bigger. And you can get all sorts of structural variants: duplications, deletions, insertions, inversions where they're switched around, translocations where they move somewhere else. And we also have copy number variants. So that is where, within the genetic code, it's not just a sequence that's spelling out different genes. You also get these regions of DNA which are highly repetitive, and the number of repeats is associated with disease. So if you have too few repeats or too many repeats, it's associated with disease. All of these things, if you're thinking about a specific genetic problem, you can go and look at how they might be inherited, and it's part of what they will kind of look at when they're interpreting the genetic mutations that they find. So talking about your genetic testing on NICU, I'm going to whiz through this because I think the clinical bit is probably the most interesting. But if you do a karyotype, you're looking at the chromosomes in a very gross way. You're looking at, do you have the right amount of chromosomes? Is the right amount of genetic material there? So if you have something where you have copies of whole chromosomes like trisomy 21, 13, 18, you can do a karyotype. It takes weeks to get a karyotype back because you have to get cells, culture them, grow them all, and then extract the chromosomes and look at them under the microscope. So about 6 weeks to get a karyotype back. A QF-PCR is a much quicker way to look at aneuploidies, because obviously a diagnosis of T21, 13, 18 is something we want to get back quickly for families, particularly if they're making decisions, reproductive decisions during pregnancy. So what you are essentially doing with that is putting in probes that specifically look for how much of chromosome 21 material is here. And if there's too much of it, if there's more than a control, it will say, yes, you have extra chromosome 21 here. This is in keeping with a trisomy. But you have to wait for the karyotype to kind of understand how that's all worked out, because there are obviously different ways that the trisomy 21 can result in different genetic variants. But it will give you a quick answer within 72 hours, so you can go back to the parents with that information. A microarray. So what a microarray is doing a whole genome-wide screen for copy number variants that are above 100 kilobase pairs. Again, it's a kind of quantitative result, so it kind of scans the genome for these copy number variants, but these are structural errors, so they're bigger. They're not looking at any problems on a single nucleotide variant level. And you compare what fluorescence is produced between patient and control DNA. So you code the patient DNA as green, for example, the control as red. And if you have the same amount of copy number variants in those regions that it's scanning for, that will be yellow because it's red and green mixed. If there's more patients, so if there's duplicated areas within the patient and they have more of that genetic material, it'll be green because they were coded green. And if there's more of the control, it will appear red because there's more of the red. So that means that there's a deletion in the patient of these copy number variants. So it misses any of these small anomalies that aren't copy number variants. And it's just quantitative. It doesn't tell you about where those mistakes are, which is different to whole genome sequencing. So it's a common first-line test that picks up these microdeletion duplication syndromes. Things like DeGeorge and Williams syndromes are types of things that you will pick up, but you will not pick up all genetic variation on a microarray. And then there are much more focused genetic testing options where you might look at a single gene. So that's things like, if you are thinking about cystic fibrosis, you would interrogate the cystic fibrosis gene genetically. If you're thinking about SMA, a gene panel is where you have a common diagnosis or a common clinical presentation which is associated with lots of genes, so they'll look at lots of genes on the panel, so like an epilepsy panel, for example, or a floppy baby panel. And then you get whole exome and whole genome technologies, and that's where you're actually spelling out the genetic code of the individual, and you have that whole sequencing information available to you. And even with kind of panels and things, we now do a whole lot more of the next generation sequencing technologies than we ever did before, and because we're able to look at this huge amount of genetic information and spell it out all at once. It generates a huge amount of data. Results are pretty complex, which we're going to talk about after, and it's like looking for a needle in a haystack amongst all of that information. So there are downsides, and essentially what we need to do, if we are doing these next generation technologies where we're spelling out the entirety of the genome or the entirety of the exome, if we're just looking at coding regions, whole of the genome, if we're doing whole genome sequencing, everything is sequenced, everything is spelt, but we don't interpret it all. We need to cancel out some of that white noise, because like I said, we all have hundreds of thousands of these genetic variants which are completely normal. So we can do different things to focus our interpretation. We can spell everything out and then put a panel on top. So we only look at the specific genes that relate to the phenotype that we're seeing. And actually most panels are now done like this, rather than looking at each gene individually, because the techniques to sequence a single gene, like Sanger techniques, are much more expensive, or they're becoming much more expensive than just doing the whole thing at once in a big shotgun and then just looking at the information that you need. Or you can do a trio exome. Some things are still looked at on a single level, but more and more we're using these next generation technologies. Or you can do a trio exome, which uses the parent's genetic information to look at the variation which is relevant to the affected individual. So if you've got two parents and you say you're unaffected, so any variants that your child has that aren't clinically relevant, you will have as well. If you have the same variants, we can just cancel all of those out. We don't have to look at them. What we're going to look at what is left over for your child that makes them different for you. So it's a way of cancelling white noise. Penetrance is difficult to interpret with a trio exome, because you may have a parent that has a mutation, but they're just very, very mildly affected because of the penetrance. So that is one of the complications with a trio, but it's a really good way to cancel out noise and focus at what you want to try and interpret if you've done next generation whole exome or whole genome sequencing. All variants that remain after you've cancelled out that white noise have to be interpreted, and there is a way of doing this. There is an ACMG classification which you can read more about online, or if you're interested, that's essentially what the genomics MSCs that are kind of offered helps you to do and be able to understand. And you're looking for, is that variant pathogenic, likely pathogenic, do you not know, is it likely benign or benign? And this is something that I did for my MSc, which was putting together kind of my own interpretation of all the different things that you need to look at to make that classification. But don't worry about this; it's all done for us as clinicians. But this is essentially what the lab team are doing, and then we'll come back to you with something and what they think the classification is. But there's a huge amount of challenges here. It's only based on our current understanding. So you might find a specific genetic variant and it classifies as a variant of unknown significance. But 15 years later, a huge trial has been done, and actually we now know that's pathogenic, but your patient will understand that that hasn't been relevant for them. And actually, therefore, there is probably a need that if you've had whole genome, whole exome sequencing, and it's something that is sort of affecting you lifelong, we probably should go back at regular intervals and actually reinterpret the variants that we see based on if anything has changed. So that is a bit of a challenge. It might miss mosaicism because you're only looking at the genetic material that you've extracted, and in mosaicism it's only certain cells that are affected. Variable penetrance affects trios, which we've talked about. Secondary or incidental findings are challenging. So that's a genetic finding that's unrelated to why you did the test, but it may be actionable. So when you consent families, you say, we might find something that we didn't expect to find that's unrelated to your child's clinical condition, but it may have implications on their health or their future. Would you want us to tell you if we have those findings? And they will either consent to have these unexpected findings fed back to them or decline, in which case the information stays on their file, but it's not given to families. And there's also huge implications for patients and the rest of their families. Things that you might end up uncovering, for example, include like non-paternity, which is a huge challenge. We don't feed that back directly, but it might be something that parents work out is there based on their understanding of genetics and the mode of inheritance. Or you might be finding other family members who may have a similar gene, but they don't want to know something like that if they're unaffected. So there are huge challenges here. So when you're thinking about consenting for these types of techniques, whole genome sequencing, whole exome sequencing, what do you think it is important to cover? And I've touched on some of them. Anything in the chat, Martina?
Not currently, though. I think one of the things that you did mention already was just about like the future implications, whether things are, so that there might be unexpected information that may be found. That's severe. And that also, again, as you mentioned, that things can change because we are getting more information. Yeah.
Totally. So we usually outline the test so that people know what's going to be done. We're going to take, blood samples. We're going to take parental blood samples if we're doing a trio. We send that off. We sequence and work out the spelling code of your genetic instructions, and we interpret that, and we're going to analyze it by looking at this or looking at that. We talk about what results we might get and what those situations mean. So we may be in a situation where we've done this test for a specific reason for your child, but we do not find any variants which we think are relevant to their condition. We may find a genetic reason for their condition, which is a relevant finding. We may find a genetic variant which we are unable to say if it is definitely harmless and we don't think it's contributing to your child's presentation, or if we think it may be contributing, we just don't have enough information on that. So it's an uncertain finding or an incidental finding, which is where we find something we weren't expecting to, and would you want to know? How it might implicate for other family members, what it might mean for any future pregnancies, if we do find something that is heritable, and also are they happy for their data to go to be used in research, and how we protect their data, because people are very worried about that. They worry about whether it's something that would affect insurance claims and things like that, which at the moment it isn't. So, but there are kind of challenges around that and various ethics around that. If you want to know what test that you need to send, just look at the National Genomic Test Directory. It goes through everything. So, for example, you'll recognize some of these indications because you will be sending tests for this in your normal clinical practice. You just probably won't have necessarily looked at the Genomic Test Directory to decide whether to send them. But the R26 is if you think there are clinical features suggestive of a common aneuploidy, so trisomy 13, 20, 1, 18, or a sex chromosome disorder, you send a karyotype and qFPCR, which is something we all do in clinical practice. But that is indication R26 within the Genomic Test Directory, and it says there it can be sent by paediatricians, neonatologists, community paediatricians, geneticists, so that we all know that we can just send that without asking for kind of specialist permission. R28 is if you've got a kind of group of congenital malformations or dysmorphism that you think might have a specific cause. So something like a copy number variant, you can start with a microarray. But there are some indications where you may want microarray and sequencing. So that is appropriate if you're thinking of a monogenic disorder, but it's not something that you would expect to see on a microarray. So you're not necessarily thinking of something that specific, and targeted testing like a kind of targeted microarray or a targeted review of a single gene isn't possible. So here you would do a microarray and a whole genome at the same time, either a trio or a singleton with a panel. And this can only be requested by clinical genetics. So it will tell you if you can go ahead and send it, what the indications are. The R14 is one that's sort of commonly talked about as well. So that's acutely unwell children who are likely to have a monogenic disorder. So that's something where a single gene is affected and affecting or manifesting in their presentation. So it just means separate to polygenic, where it would involve lots of genes or lifestyle issues. It's something that presents in childhood and makes you very unwell, where the phenotype suggests that, but there is no focused testing available. So if you were thinking about an acutely unwell child, like let's say a child that you think has SMA, but there is specific testing available, you would do that first. These are for the children where the diagnostic kind of net has to be much wider. And you do this as a whole genome, either trio if you don't know what you're looking for, or a panel if it's something where there's a group of genes that you can look for that are commonly seen in that presentation, and it has to be requested by genetics. And then there's other indications like achondroplasia testing, that single gene testing, hypertonic infant panels, cystic fibrosis, single gene testing, Prader-Willi testing, SMA testing. So that's all on there if you're thinking about it. In clinical practice, when you're applying this, you obviously always start with a history and examination and look at the directory, and that will help you indicate testing. If you're unsure or it's something highly specialist, just discuss with genetics. I'm going to run through some cases. I'm running out of time, so have a think about your answers, but I'm not going to wait for them. So you're the page holder on for neonates and you're asked to see a baby on postnatal wards with unusual features. What do you think about? History is really important to explore the antenatal history. You're thinking about scans. What was their combined risk score of having trisomies, essentially? So they look at blood tests and nuchal translucency in pregnancy to give you a risk whether you're high risk for aneuploidies or not. Look at growth profile, maternal health, medications, substances. So there are certain substances that present with specific clinical features. So like fetal valparate syndrome, where if you have that, you're not necessarily looking for a genetic cause. History of miscarriages, stillbirth, previous child death is really important. If there's any assisted conception, and if there is, find out where the gametes came from, who's biologically related to that child. There's no point doing a trio with an unrelated individual because the family's had assisted conception. Birth history, how, at what age, rupture of membranes. Again, if you've had long rupture of membranes, you end up with a sort of Potter's-like syndrome, and those children could be quite almost dysmorphic looking, but it's not necessarily a genetic cause. Or are you worried at birth? Family history, often there are subtle features which are just familial, so have a good look at parents, find out about anything that runs in the family and ask if they are related. It's awkward, but it's really important. And then the history of the clinical presentation, who has seen what, when was it noticed, are the parents concerned? Examine the baby head to toe, look at their growth, senior review. What does that baby have? It's a spot diagnosis. Hopefully everyone's thought of trisomy 21 because that is a typical appearance of.
We've had that answer, T21.
Yay. Brilliant. You've got epicanthic folds, you've got upslanting palpable fissures, you've got a broad nasal bridge, you've got brachiocephaly there, you've got an adorable cute baby who probably has trisomy 21. So thinking about what you would do for this patient, what genetic testing is indicated. We've talked about this already, but this would be RR26, sending off a karyotype and qPCR and making sure the parents know in 72 hours, I'm going to call you with provisional information on whether it confirms what I thought clinically. And then we'll have 6 weeks that will give us a bit more information about how this has been, occurred genetically. What other investigations are needed? Babies who have T21 need their newborn screen to look for hypothyroidism. They need a full blood count to look for myelodysplastic syndromes. They need an echo because they might have an associated cardiac lesion. You need to make sure they feed because it's associated with duodenal atresia. They need ophthalmology. They need long-term follow-up. So I'm just going to explore some things that are really important to think about with your communication for these families. It's something that you probably are aware of, but there's a bit of controversy amongst the families who have kids with T21 about using congratulations. Some families feel really upset that we go in with a very pessimistic, breaking bad news type voice. We don't say congratulations if they're completely accepting of their baby having trisomy 21. So a lot of them will say, I was really disappointed that no one congratulated me on the birth of my baby, and everyone kind of came in soft, hushed, somber tones to deliver this bad news, and I was totally in love with my baby, and it didn't matter. So I'm really against making it sound like it's such a bad thing. But for some families, it's really jarring if that's unexpected or if it is bad news for them, for us to be like, congratulations, this should be joyous. Here's the news that you don't feel great about for now. And then, it can be very difficult. So I always say, your baby's really lovely, because that is, I think most parents will think that irrespective of where they fall on that spectrum of not being bothered to it being very worrying. They still know their baby is lovely and they still have, a lot of care and love for their baby. It's just coming to terms with this diagnosis. I find that works really well. Finding out what parents know. So I've been asked to come and see you. Were you expecting me? Do you know why I've been asked to come? Often it's no, because what seems to happen in lots of my experiences, the midwives can panic sometimes when they see these babies. They pull in an SHO, they panic, and then you're asked to come, and this kind of situation has evolved. So I always say, even if a parent says, no, I have no idea why you're here, I say, is there anything that you've noticed about your baby that is different or worries you? And most times parents will say, yes, the last one that I did, she said, yeah, my baby has Down syndrome. I know, I can see it. Everyone's kind of been running around and sort of treating me like I might break. So it is really important that you kind of explore that. Examine the baby and then give the baby to the parents, especially if it's unexpected. So you've examined to look for all your features you're then going to discuss, and have them holding the baby. I think that's really powerful, especially if you're breaking bad news. Explain what you found, what you think it means, and you can frame the uncertainty depending on the situation. So there are some babies who are really barn door, who you'll say, I think this is Down syndrome. You'll explain all of that because of X, Y, and Z features. I can't be 100% sure until I've got these results back, which I would expect a provisional diagnosis in 72 hours. Or there might be just very subtle features and you think this is really unlikely, and actually mum has got epicanthic folds, and I think I still need to do the test because I'm not 100%, but I think it's less likely. You can frame that still within that spectrum, but slightly different slants depending on where you lie on that spectrum, and then let them know what's going to happen next with clear timeframes. I'm going to quickly run through case 2, and case 3 is short, but imagine a term baby who had a background of polyhydramnios, who was very floppy at birth, but breathe normally, so comes to NICU. You do the same history, looking at all of those things. You examine the baby, and that includes a neurological examination, and whether they're well or unwell, because a floppy baby will be one or the other. You're thinking about whether this could be central. So in the acute phase, these babies can be weak. So HIE is a central cause of a floppy baby. So that means it's affecting the brain or the spinal cord. But as it goes onwards, they tend to have normal strength in the non-acute phase. You're looking for dysmorphic features. You're looking for brisk reflexes, irritable, and a history that fits with something central. Peripheral, these kind of babies more with like muscular and neuropathies. So they have reduced strength, reduced reflexes, muscle fasciculation, myopathic phase, weak cry. So you're looking at where the baby comes on that spectrum. And the causes can be in any of those areas. So it is wide ranging, the kind of causes of a floppy baby. You will have a guideline. Almost everywhere has a guideline on how you investigate a floppy baby. So you're doing baseline metabolic tests, you're checking for infection, you're doing baseline biochemical tests, looking at cranial ultrasound. There may be specialist tests that you need, but genetic testing will be on there in your approach to a generically floppy baby, unless they have an obvious cause like HIE and you know it's HIE. If you think it's peripheral, you're doing genetic testing for SMA, Prader-Willi, and the muscular dystrophy panel. This is what my local guideline says. But if it's central, it's much more, it's a wider net that you cast, and you would do a microarray and a whole genome sequencing. But imagine this baby who we found, we've done the genetic testing, we think it's peripheral, and we find a pathogenic mutation in the SMN1 gene, and we diagnose SMA. So spinal muscular atrophy happens because of mutations in the SMN1 protein. So essentially without that protein, which is called the survival motor neuron protein, the motor bodies in the anterior horn cells, the motor neurons can't operate optimally. But you have another gene called SMN2, which produces essentially a less good survival motor neuroprotein. And if you have lots of copies of that, you'll be affected less severely because it compensates for the absence of SMN1 to some extent. If you have few copies, it's more severe, and that accounts for the variety of clinical phenotypes that you see. But if we diagnose this early in this patient, they may be eligible for novel gene therapies, which is what's really important here. So Zolemska is an antiviral treatment therapy. So you put the SMN1 gene into a virus and hope to integrate it into the patient genome. So that's being developed. And nusinersen, which you've probably heard about. So this is an antisense nucleotide. So it's not changing the genetic code, but it's influencing how SMN2 is processed, and it's forcing it to keep in the more important functional domains that make SMN1 work and SMN2 not work so well. It forces it to keep them in, so you get more functional protein essentially. And this is something that we are offering as a treatment now. It has its limitations. You're only eligible depending on where you're diagnosed and what your function is like, and you have to get 4 monthly intrathecal injections because you're not changing the genetic code, you're changing how the protein works. You need to keep giving it regularly because you're altering RNA, you're not changing the genetics. And then I'm just going to blast through this really quickly. This is a case that we had here recently. So it was a 28-week baby who had a relatively smooth neonatal course, but even though they got older, they kept having pauses in their breathing despite increasing maturity. So history and examination always. But the progression for this baby is that we optimize their non-invasive respiratory support. They kept needing to be ventilated because of these significant apneas. We optimized their caffeine. We screened them for sepsis every time. Didn't turn out to be sepsis. They had a normal echo. We treated reflux. We thought about seizures and did an EEG. And we did an MLB to check that there wasn't a vascular ring or anything, all of which was normal. So we asked genetics to see them. There was a bit of a family history of hearing impairment. Mum had hypermobility and dad had had an inguinal hernia. But the baby did have some subtle dysmorphic features. So bilateral talipes, inguinal hernias, hyperextendable fingers, downward slanting palpable fissures, although they weren't sure because he was sort of quite a small, ex-prem looking baby. Very upturned nose with large nostrils, which they again weren't sure if that was because of CPAP or part of the baby's phenotype, and a small pointed chin. So they did a microRNA qPCR to start with. And then if the microRNA, the qPCR was negative, they were going to upgrade to an urgent trio exome, which is exactly what they did. They didn't want to wait 6 weeks for the karyotype. If the qPCR was normal, they were happy that was enough to go through. We talked about FOX2B testing, which is associated with congenital hyperventilation syndrome, which is what we all thought clinically we might be looking at because of these significant pauses in breathing. They were very significant apneas, but because of all these clinical features, the decision was made that we actually needed to look wider, and we'd catch it if we did whole genome testing, and we'd look for other things too. And they ended up finding a pathogenic de novo mutation in the NALCN gene, and this causes this presentation: congenital limb face contractures, hypertonia, developmental delay, which presents exactly as this baby presents. So it's been really important for this family because we now know a bit about more what to expect from him. We know that we're not going to get rid of these apneas completely. We can think about his long-term planning, and that means that we can progress things much more. There's still a lot of uncertainty because some children are only mildly affected and the phenotype is very severe for others, but we've been able to explore what they will do at home, what they think about seeing of care, and the family has been able to speak to other families who are affected, which is really important. So that's all been amazing. And essentially the future of genomic medicine is very much still developing. If you're interested, have a look at the Newborn Genomes Programme. I'm not going to talk about it because we all need to get you away, but we're now going to be doing an evaluation of what genetic screening for 100,000 newborn babies looks like, and the clinical utility that may have diagnostically, but also later in their life. So it's really exciting. It's a rapidly evolving specialty. Next generation technologies are used increasingly. We have resources to guide our genetic testing, so always look at that when you're thinking about doing genetic testing. It will often give you the answer, and there's a huge potential for growing research in this area. And that is your code. Does anyone want to ask me anything?
Thank you so much, Jess.
No worries.
I think those last bits, those like key summaries and those cases, knowing that we have a national genomic test directory to go to if we're not quite sure, as well as those examples that you had when it comes to communicating with families about their babies.
I can send this to you, Martina, and it has all my notes at the bottom. So if there's anything I haven't said, because we did rush through it a little bit at the end, people can have a little read through those notes if they're interested. And yeah, if anyone's interested in developing a subspecialty interest and wants to know more about what that looks like, you're very welcome to email me as well.
That's amazing. I mean, we don't have any questions in the chat right now. But if anyone does have anything, please do pop it in there. If not, can you please just register your attendance, and there should be an email sent out for you to complete a survey. So that would be excellent. And I think it is just sort of bringing it back to the start where you started, Jess. It's so interesting how actually we do use so much genomic medicine in our daily practice, regardless of what specialty we're working in. We're sending off PCR tests for all sorts of things all the time, I'm sure, especially right now that it's winter, and just the sort of really wide-ranging implications and applications of it.
Yeah, it's really cool. And it was, I mean, I'm not going to use this this MSc a huge amount, if I'm totally honest, in clinical practice, but it was so lovely understanding at the micro level that you do when you're doing something like a MSc. So it's been really enjoyable, even just to like learn more about the science, even if it's going to be different in clinical practice.
Okay, we don't have.
Could I please ask a question?
Yeah.
Yeah.
Thanks. Sorry, I can't seem to access the chat. Apologies. I just wanted to ask if you could just explain, because we've had a situation recently where we've done a whole genome sequencing on the baby, and then they asked us to send the parents a blood test. Sorry, just to be clarifying the times when you need the parental sample and when you don't. That would be really helpful. Thank you.
So it depends on if you're going to do a panel. It depends on the indication, but essentially, if you're sequencing the whole genome, you just end up with this huge spelling of your genomic book, like so much information. And there's variants all along the way there, like hundreds of thousands of variants in any individual. And most of those will be normal. So you can either cancel out all that information that you don't need on those variants, which are completely normal, in two ways. If you have a specific indication, so if you have a floppy baby or an epilepsy, for example, and you just want to say, well, this is epilepsy, I want to do an epilepsy panel, I'm going to look at the 40 genes that are associated with epilepsy, or however many there are, and you can look at something called PanelApp, which will tell you what the common panels are. So you can get that whole genome sequence, because it's going to be cheaper than individually sequencing 40 genes by doing it with this massive parallel sequencing technique. But you're then going to look at whatever genes are in your panel, so let's say 40, and you've looked at the variants and you've tried to interpret them, and either you end up with too many variants, let's say, for you to interpret, or you don't end up with anything diagnostic. The other way to then, because you've still got all that sequencing data is still there, it's sitting there for you to explore if you didn't get the information on the first line, or if it can't cancel out, for example, enough white noise on the first line, although I don't know how often that actually happens. But if you need to cancel out the information further, test the parents, because any variant that you've inherited that's just normal variation, you both have it, you're both, they're unaffected. So any variant that they have that you also have, you would not expect to cause disease. The only caveat to that is if there's variable penetrance, but that's rarer. So you then do the whole of the parent's genetic sequencing, and any of the variants that are found in you plus a parent, you don't have to look at. Just say, well, it's normal because the parent is normal, so it's not causing the disease because only the child is affected. And then look at what's left over, what variants are unique to your patient. So most testing, apart from single gene testing, is now done with these next generation technologies, and you have to go and work out what you want to look at, because it's just way too much information. You can't look at all of it, in two ways: either apply a panel or look at the parents trio, and you might end up with one or the other depending on what you've done before. Does that make sense?
Thank you. That's really helpful. Thanks.
Cool.
Perfect. I think we will say goodbye then. Thank you everyone for joining, and thank you, Jess, again for your time and for teaching us.
No worries. Sorry about the helicopter. We're by the helipad, so it goes off sometimes.
That's okay. Brilliant.
Bye. I'll see you later.
Bye.
Hello,
thank you.